If you follow longevity research at all, you're familiar with the usual suspects for tracking aging. We're talking biomarkers.
Right, things like telomere shortening or somatic mutations building up, and of course the big one lately, DNA methylation.
The basis for all our epigenetic clocks.
Yeah.
And there's always been this fascinating pattern.
That the rate of change in these markers seems to be linked to how long a species can live.
Exactly.
Yeah.
Short-lived animals, the changes happen fast. Long-lived ones, they change incredibly slowly.
It looks like a fundamental law of biology.
But here's the question that really drives our deep dive today. Is that link a profound biological truth, or is it, just a mathematical trick?
A trick that comes from how we define the word rate.
How so?
Well, think about it. If you calculate anything as change per year, and then you compare a mouse that lives 2 years to a whale that lives 200.
The very act of dividing by years is going to make the whale's rate look smaller.
Precisely. It's baked into the math. It could just be an arithmetic artifact, not a real discovery about aging itself.
So how do we get around that? How do we prove the link is real?
That's what we're digging into. A new analytical framework designed specifically to isolate the true biological signal and get rid of that statistical bias.
And the data for this is pretty incredible.
It's massive. The sources draw on the Mammalian Methylation Consortium. We're talking 90 different dog breeds and over 125 mammalian species.
Everything from a shrew to a bowhead whale.
Basically, yeah.
So our mission today is to find the real bias-free equations that connect this average rate of change in methylation, the AROCM, to maximum lifespan. And, maybe more importantly, to find out where in the genome this is all happening. Okay, let's start there then. Before we even get into the statistical fix, where is the biological action? Finding the right genomic spot feels like the real bombshell here.
Absolutely. The math doesn't mean anything if you're looking in the wrong place. They looked at, I think, 54 different chromatin states, different ways DNA is packaged.
And the signal wasn't just everywhere. It was specific.
Very specific. It was highly localized. They zeroed in on the methylation sites, the CPGs, that are located in what are called bivalent promoter 2 regions.
BivProm2 plus Klaska. Okay, and why there? What's special about those regions?
This is the key. These BivProm2 plus regions are strongly associated with the polycomb repressive complex 2.
PRC2, right? That's a master regulator, isn't it? It's famous for gene silencing, maintaining cell identity, especially in stem cells.
It's the ultimate epigenetic gatekeeper. It's crucial during development for keeping very important genes turned off until you absolutely need them.
So the idea is that aging is linked to the decay of this fundamental stability system.
That's what the data is pointing to. The rate of change in these specific regions is the best predictor of lifespan. It's about losing that core developmental stability.
Okay, so that gives us the biological target. Now let's go back to the math, the statistics, because even measuring the rate there was flawed at first.
Right, the simple average rate of change in methylation, or AROCM, it wasn't enough. They had to create an adjusted AROCM, an AJ AROCM.
And why the adjustment? You mentioned something about unequal sampling.
Yeah, I mean, when you're gathering samples from 125 species in the wild, you can't just order them up by age.
You get what you get.
You get what you get. So for some species, you might have mostly young animals. For others, maybe a lot of really old ones. This creates a skew, a bias in the data.
And they measure that skew with something called the standard deviation of relative age.
Yeah.
SDR.
Exactly. It's a measure of how spread out your samples are across the lifespan.
Yeah.
If your SDR is low, it means your samples are all clustered together and your calculated rate is going to be inaccurate.
Because if you only measure the rapid changes in youth, your rate will look way too fast.
Precisely. The AGA-day ROCM uses a statistical fix based on that SDR value to correct for those sampling flaws. It basically calibrates the rate.
And when they applied that fix, what happened to the signal? The dog breeds are the best example.
Oh, it was night and day. In the 90 dog breeds, the unadjusted, noisy AROCM had a correlation with lifespan of negative 0.36.
Which is kind A.
Yeah.
It's there, but it's not very strong.
Not at all. But once they applied the adjustment, the correlation jumped dramatically to negative 0.75.
Wow. That's an incredible cleanup. You're not just finding a pattern. You're revealing one that was buried in the noise.
And it gets even better. When they took this adjusted model and applied it to all 125 mammal species, the relationship became almost a perfect law.
The big aha moment.
The big one.
Mm-hmm.
They found the adjusted AROCM is almost the exact reciprocal of a species' maximum lifespan.
So AGAR-OCM is basically 1 divided by lifespan.
Yes. The correlation there was, get this, 0.96. It's almost a perfect fit.
So the rate of decay in these specific polycomb regions isn't just related to lifespan.
It seems to be the fundamental constant setting the speed limit for that species.
Okay, if that rate is the speed limit, we have to talk about what isn't, because
Yep.
that was another huge and frankly really counterintuitive finding here.
Yes, so we've established the rate is what matters. But what about the overall strength of the epigenetic clock?
You mean, how well does methylation actually track with chronological age in a given species? That seems like it should be hugely important for longevity.
You would think so. But the study found that the correlation between methylation and age, they call it core methyl age, has almost no relation to maximum lifespan.
Wait, really? None?
Practically none. It's amazing. You look at a long-lived species like us, humans, we have a median correlation of 0.51. Humpback whales are at 0.58.
Okay.
Those numbers are right in the same ballpark as a short-lived mouse at 0.66 or a brown rat at 0.67.
That is just, it's stunning. So it's like every mammal has a perfectly good watch. The watch's accuracy isn't the issue.
That's a perfect way to put it.
The issue is how fast the battery in that watch runs down. And that's the AROCM.
The analogy is perfect. The strong link between methylation and age, the clock itself, is basically conserved across all mammals. That's why we can build these universal epigenetic clocks. But the thing that sets the ultimate boundary on life isn't the clock's general accuracy. It's the specific rate of decline in those BIV-PROM2+ regions.
So the universal clock is the speedometer. But the AROCM in those specific regions is the engine block that determines the car's top speed.
I like that, yes.
Okay, so let's talk about that rate over time. Is it constant? Is the AROCM the same when we're young versus when we're old?
No, the data shows it's often nonlinear. You can see it pretty clearly in some species, like the green monkey or the beluga whale. The line charting methylation against age has a curve to it.
It's not a perfectly straight line, so how do they handle that?
They divided the lifespan into 2 phases based on relative age, which they call R. It's just your age divided by your species' maximum lifespan.
Simple enough.
They defined young as a relative age of less than 0.1. So for a human with a max lifespan of 122, that's everything before age 12, basically your developmental years. Old was everything after that.
And what happened when they compared the rate in that early developmental phase to the rate in the much longer old phase?
This was a huge finding. They found a really strong positive correlation over 0.78 between the AROCM in young animals and the AROCM in old animals.
So the rates are proportional.
Exactly. The absolute rate is faster when your young development is happening fast, but the key is that the rates are proportional. AROCM young is proportional to AROCM old.
What's the biological meaning of that? Why is that proportionality so important?
It points to a single continuous mechanism. It suggests a species is using the same fundamental epigenetic maintenance system from day one all the way into old age.
So it's not like 2 different processes for growing up and then growing old.
Right. If aging were just random damage, or if the early life and late life mechanisms were totally separate, you just wouldn't see that tight, predictable link. The speed limit that's set early on seems to dictate the speed limit for the rest of your life.
Okay, before we get to what this all means for us, we have to talk about the confounders, the statistical due diligence.
Of course.
The elephant in the room is, well, literally the elephant. Body size. Big animals tend to live longer. Did they account for that?
They did. They ran a multivariate analysis, which lets you test if one variable is just a stand-in for another.
So is AROCM just another way of saying body size?
And the answer was a clear no. When they put average adult weight into the model, it had a weak, non-significant effect. The p-value was 0.45, which means it wasn't a real predictor once AROCM was in the mix.
The epigenetic rate is the real driver.
It's the independent, powerful predictor. The body size effect almost completely vanished.
So it doesn't matter if you're a Chihuahua or a Great Dane. The rate in your polycomb regions predicts your lifespan. That is incredibly clean. What about tissue type?
They checked that too. Most tissues that divide a lot, they all showed similar correlations. Tissues that don't, like the brain, had slightly lower correlation.
Any outliers?
The liver, actually. It was the only tissue that, after controlling for everything else, still showed a significant positive association with the AROCM.
Interesting. So the liver might have a slightly unique aging rate?
It might. But for the most part, the main finding holds up across different tissues.
And the last big one, shared ancestry, phylogeny. Did they make sure they weren't just seeing patterns because, monkeys are similar to other monkeys?
Yep, they adjusted for that too. Even after they stripped out the influence of shared evolutionary history, the inverse correlation was still there. It held strong.
Okay, so this finding seems incredibly robust. It holds up across body sizes, tissues, and the tree of life.
Hmm.
We have this clean equation. The adjusted rate of epigenetic change in these BIFCOMPROM2 plus regions is the inverse of lifespan.
That's the core finding.
So what does that mean for you, the listener? What does having this quantitative link actually open up for aging research?
It turns a huge biological mystery into a number you can measure, a target. If we know the equation for the speed limit, we now know exactly what we need to slow down.
It gives you precision,
Hmm.
which leads to this idea of the peak span.
Exactly. The argument is that to really advance restorative therapies, we should focus on what they call the near peak edge.
That's the 90 to 100% productive lifespan range.
Right.
Why focus so narrowly there? Why not just try to fix things when someone is 70 or 80?
Because that's where a small change in that fundamental rate of decline, the AGR-ROCM that we can now measure, gives you the biggest possible return on investment.
Compounding returns.
Compounding returns. If you can just slightly reduce the slope of that decline when your function is still really high, you get exponentially more healthy, productive years on the back end.
And this gives us a way to test if a therapy is working on a mechanistic level. You don't have to wait 20 years for a clinical outcome.
Exactly. You can just ask, did this intervention lower the AROCM? If it works, the rate has to go down. It moves the conversation from just clocking age to actually understanding and hopefully controlling the speed of aging.
So this deep dive really gives us a fundamental equation for life's speed limit. The aging rate, the AGR-OCM, in these very specific genomic regions is almost exactly 1 divided by a mammal's maximum lifespan.
A truly foundational finding.
So here's a provocative thought for you to take away. If the rate of aging we see in our earliest developmental years is fundamentally proportional to the rate we experience in old age, and if that rate is mathematically locked to our ultimate lifespan, what does that really imply about the power of early life interventions? Does slowing that core AROCM when we're young change the entire trajectory far more powerfully than any rejuvenation strategy we could try later on? And how can we now use this incredible new measurement to finally put that question to the test?